Types of microscopes. Since microbes cannot be seen with naked eyes, microscopes provide magnification which enables one to see them. It is one of the greatest tools of a microbiologist. Hence, it is important to understand the types of microscopes and their applications. The first microscope was developed in 1590 by Dutch lens grinder Hans and Zacharias Janssen. Antony von Leeuwenhoek was the first person to observe living cells under the light microscope in 1675. Today's microscopes look quite different than those built by Leeuwenhoek, allowing higher magnification and clearer images. Some of the most common types of microscopes used to study microorganisms are categorized as number 1, light microscope, and number 2, electron microscope. Light microscope. These microscopes use a beam of light to create an image. Magnification is obtained by a system of optical lenses. Various types of light microscopes include bright field microscope, dark field microscope, fluorescence microscope, phase contrast microscope, and ultraviolet microscope. Bright field microscope. It is the most basic type of microscope used in microbiological labs. It uses visible light and multiple lenses to magnify a specimen. The object being studied appears darker on a brighter background. While it is most widely employed for routine microscopic work, its resolution is limited. Dark field microscope. In this microscope, only light scattered by specimen enters the objective lens. It creates a bright image of a microorganism against dark background. This is accomplished by equipping a light microscope with a special kind of condenser known as Eppes condenser. Dark field microscopy is particularly valuable for examination of living unstained cells or microorganisms suspended in fluid, that is wet, and hanging drop preparation. Fluorescence microscope. This microscope uses fluorescent dyes to label specific structures or molecules within a microorganism, allowing one to visualize them. When observed under light microscope with UV illumination, organisms that are stained with fluorescent dye will appear as luminous objects. Fluorescent dyes, when chemically combined with antibodies, make it possible to identify individual cells that reacts with these labeled antibodies. This technique is known as fluorescent antibody technique or immunofluorescence. Phase contrast microscope. It is extremely valuable for studying living cells, microbiology, microbial motility, endospores, and is widely used in applied and biological studies. It converts light differences in refractive index and cell density into easily detected variation in light intensity. It employs controlled illumination of a specimen accomplished by special phase contrast objective and condenser assembly attached to a conventional light microscope. Its condenser has an annular stop, an opaque disc with thin transparent ring that creates a hollow cone of light. Controlled illumination make it possible to distinguish structural details that vary only slightly in thickness and refractive index. Ultraviolet microscope. Ultraviolet microscope permits greater resolution and greater useful magnification than that obtained with conventional light microscope. The characteristic absorption of ultraviolet rays by certain substances permit their localization, distinction, and measurement in suitable specimens even in a living state. The images are recorded on a photographic plate. Preparation and staining of specimen for light microscopy. Fixation and staining increases the visibility of microorganisms, accentuates specific morphological features, and preserve them for future study. Fixation is a process by which the internal and external structures of specimens are preserved and fixed in position. It inactivates the enzyme that might disrupt cell morphology and toughens cell structures. Two fundamentally different fixation procedures are heat fixation and chemical fixation. Heat fixation preserves overall morphology by inactivating the enzymes. Chemical fixation is used to protect the fine cellular substructures as well as morphology of larger and more delicate microorganisms. Common fixative mixtures contain ethanol, formaldehyde, glutaldehyde, etc. Dyes are used for simple or differential staining procedures. They have chromophore group that gives dye its color and bind cells with ionic covalent or hydrophobic bonding. These are of two types, basic dye and acidic dye. Basic dyes. These are methylene blue, crystal violet, safranin, malachite green, basic fuchsian. They bind to negatively charged cell molecules like nucleic acids, proteins, surfaces of bacterial cells. Acidic dyes include eosin, rose bengal, and acid fuchsian. They bind to positively charged cell structures. Dyes that bind through covalent bonds are also useful. Examples: Sudan black that stains lipid and shift reagents for staining DNA. Number 2 is electron microscope. These microscopes use beam of electrons instead of light to create an image. Electron microscope. These microscopes offer much higher resolution of roughly 1000 times than that of light microscope, hence allowing visualization of fine details of microbial structures. There are two main types of electron microscopes: transmission electron microscope and scanning electron microscope. Transmission electron microscope. A heated tungsten filament in the electron gun generates a beam of electrons that is focused on a specimen by a condenser. Since electrons cannot pass through a glass lens, donut-shaped electromagnets, its magnetic lenses, are used to focus the beam. The column containing the lenses and specimens must be under high vacuum to obtain the clear image, as electrons are deflected by collision with air molecule. The image can be captured digitally or in photographic film as a permanent record. Preparation of specimens. Specimens are prepared for observation by soaking thin sections, that is 20 to 100 nanometer, with solutions of heavy metal salts such as lead citrate and uranyl acetate. Osmium tetroxide fixative also stains specimen and increases their contrast. The stained thin sections are then mounted on tiny copper grate and viewed. Other important techniques for preparation of specimens are negative staining and shadowing. In negative staining, the specimen is spread out in a thin film with either phototungstic acid or uranyl acetate. Heavy metals render background dark, whereas the specimen appears bright in a photograph. Negative staining is an excellent way to study the structure of viruses, bacterial gas vacuoles, and similar objects. In shadowing, a specimen is coated with a thin film of platinum and other heavy metals by evaporation at an angle of 45 degrees from horizontal, so that the metal strikes the microorganism on only one side. This technique is particularly useful in studying virus morphology, bacteria, and archaeal flagella and DNA. The shapes of organelles within the cell can be observed by transmission electron microscope if specimens are prepared by freeze etching procedure. The cells are rapidly frozen in liquid nitrogen. They become very brittle and break along the lines of greatest weakness. The exposed surfaces are then shadowed and coated with layers of platinum and carbon to form a replica of the surface. This replica is studied in transmission electron microscope, providing a detailed three-dimensional view of intracellular structure. Scanning electron microscope. Scanning electron microscope scans the beam of electrons over the surface of a specimen. It produces an image from electrons released from atoms on a specimen's surface. It has been used to examine the external surface features of microorganisms in great detail. We can get a three-dimensional image of a microorganism's surface. Thank you for watching. Don't forget to subscribe and hit the notification bell to remain updated.